Hi everybody. What I want to talk about today is the steps of synaptic transmission. So what happens at a synapse, and I'll also talk a little bit about integration. So what happens in the postsynaptic cell after the neurotransmitter arrives. So first you have to know where we are in the nervous system. So if you don't know about neurons, go back and review that, keeping in mind that the dendrites are the receiving area of a neuron, and the cell body integrates the signal, and we'll see that at the end here. And the axon's job is to carry action potentials potentially over a long distance. Some axons are short and some are long. And at the end of the axon, there is the axon terminal, which is the point at which the synapse exists. So the meeting of the axon terminal of that first cell, which we'll now call the presynaptic cell, with a dendrite, or sometimes the cell body, of the postsynaptic cell, the second cell in the circuit. This is an actual picture of a synapse. The color is not real, but the rest of the picture is there. So at the axon terminal, we have mitochondria, of course, for energy, but the main thing I want you to see here is that vesicles filled with neurotransmitter are already present. So they've moved slowly through slow axonal transport down the axon over time, and are essentially waiting for an action potential to arrive. Then looking a little more schematically, we can see that what happens when an action potential does arrive at the axon terminal is that instead, so normally as an action potential moves, it starts a new action potential by opening voltage-gated sodium channels. At the axon terminal, there aren't any voltage-gated sodium channels. Instead, we have voltage-gated calcium channels, shown here. Because calcium is pumped out of cells, and because the inside of the cell is negatively charged, once a calcium channel opens, calcium will enter the cell down its electrochemical gradient. So the channel's opened by the change in voltage, the calcium goes into the cell, and through a series of binding events of calcium-dependent proteins, excitatory of the neurotransmitter occurs. That is, those vesicles of neurotransmitter that had already been present at the synapse are released into the synaptic cleft. So the actual space between these two cells is called the synaptic cleft, and it's very small, 30 nanometers or sometimes even less, a very small distance between these two cells. Because the distance is so small, diffusion of neurotransmitter across the synaptic cleft is quite short. In other words, it goes very quickly, and quite soon after the action potential has arrived in the presynaptic cell, the neurotransmitter arrives at the postsynaptic cell. Now, what happens at that point depends on what kind of receptor is present. So let's look at the options here. A common option is what is sometimes called an ionotropic receptor, where the neurotransmitter binds to an ion channel. In other words, the receptor for the neurotransmitter is also an ion channel, and the ion channel changes conformation when it's bound by the neurotransmitter and opens. This could be an ion channel for a variety of ions, but most commonly it's either a sodium ion channel, a potassium ion channel, or sometimes an ion channel that can pass both sodium and potassium. Chloride channels are the final major option that's possible. So that's one set of possibilities, that an ion channel will open and then an ion will move down its electrochemical gradient. For example, sodium moving down its electrochemical gradient into the cell, or potassium out. Another option is these, what are sometimes called metabotropic receptors, which are simply G-protein coupled receptors that you may be familiar with in other contexts. The receptor is bound by the neurotransmitter, which activates a G-protein, and that can either lead to immediately opening and closing of ion channels, or sometimes some other kind of cellular response, for example, a change in transcription or translation, the phosphorylation state of an enzyme, or something else. Most commonly, the result of a neurotransmitter arriving at its postsynaptic cell is to open or close ion channels, and most commonly to open them. So if ion channels open, whether triggered by an ionotropic or a metabotropic receptor, and these are sometimes now called simply fast and slow postsynaptic responses, then what you have is ions moving in or out, and therefore charge is moving in or out, and therefore the voltage of the postsynaptic cell, the membrane potential of that postsynaptic cell, is changing. And it can only change in two ways, right? It can either become more polarized. It starts out about minus 70 millivolts, so it could become more negative. And that would happen if a positively charged ion left the cell, or a negatively charged ion like chloride entered the cell. Or it could become less polarized. It could get closer to zero. And that would happen if a positively charged ion entered the cell, for example, sodium. If the result is depolarization, going closer to zero, then we call that an excitatory postsynaptic potential. So it's postsynaptic, right, because this is the cell post or after the synapse. It's a potential because it's an electrical change. And it's excitatory because if the cell becomes relatively depolarized, that will push the cell towards the threshold for its own action potential. It's not the case that one EPSP will lead to an action potential. There are a couple special cases where that is true, like in skeletal muscle, but in most postsynaptic cells, it's not true. You would need multiple EPSPs to add up, and we'll see the adding in a minute, to the threshold. But the other possibility, right, is that the cell became more polarized, hyperpolarized, and we call that an IPSP, or inhibitory postsynaptic potential. The reason why it's inhibitory is that you're making the cell even more negative and therefore taking it farther from the threshold voltage. You're making that cell less likely to have an action potential in the near future. Let's look at these postsynaptic potentials. How does the cell sort of add them up? First, you have to remember that it's not the case, as we always see in pictures and textbooks, that a synapse is always shown with one presynaptic cell and one postsynaptic cell. And that is what a synapse is. But most postsynaptic cells don't just have that one synapse. They have many different neurons, the axons of many different neurons converging on their dendrites and cell body. This isn't always the case, but especially in the central nervous system, it is often the case. So this is a picture of a cell body and its dendrites, these extensions here, these short extensions, and then these are all terminals, axon terminals of axons that are synapsing on it. In fact, there are individual neurons in your brain with more than 10,000 synapses, that is 10,000 axon terminals arriving at that cell. In other cases, there might be only 3 or 4, but there's almost always some kind of adding up that has to be done with different cells. So if you imagine then, if you imagine this blue cell here is the postsynaptic cell, and the little extensions here are these little dendrites coming off of it. So in this example, we have 2 excitatory inputs, that is, cells that are going to release excitatory transmitters, that is, ones that are going to depolarize the cell to produce EPSPs, and 1 inhibitory neuron. So these 2 are going to tend to make the cell a little more positive, little depolarized, and this one's going to make it a little more negative. And in this example, there's not enough total positive charge. You know, it's like +1, +1, -1. So you end up with a net depolarization, but not enough to get the cell to threshold, to about -55 millivolts, the point at which the cell would have its own action potential. The key location is what's labeled here as the trigger zone, sometimes called the axon hillock, which means the very beginning of the axon. It's only in the axon that you're going to be able to trigger these action potentials. So once these ions diffuse through the cell body, is there still enough positive charge here to get an action potential? In this case, no. The inhibitory neuron was enough to cancel out the input from those other two. So the cell can add these up sort of in two different ways, in time or in space. So let me explain what I mean. Spatial summation means imagine that the postsynaptic cell was receiving a lot of inputs at the same time, all right? And each individual one of these positive inputs, these EPSPs, is not strong enough to take the cell to threshold, but adding them up is enough. In the last one, we had 2 positives and 1 negative, right? 1 inhibitory. These are all excitatory. And if these 3 excitatory neurons fire at about the same time, their positive charge before it dissipates will add up potentially to the threshold, and then you get an action potential here. These are these postsynaptic, these graded potentials, but the sum of them, if it's enough to reach minus 55, will trigger an action potential. Another possibility, so you don't actually require multiple neurons to ever have an action potential. It is possible that you could have one input or just a few, but that it was repeatedly, in a short period of time, stimulating that postsynaptic cell. So for example, if this cell with this little dendrite here on this cell body had 2 releases of neurotransmitter in a short period of time, triggering 2 postsynaptic potentials, if they were separated in time, you can see that if you get back to your baseline membrane potential, then the second one's not going to add up. But if the second one comes while the first one is still depolarizing that cell, then they can add up. And remember, action potentials never do this. It's impossible for action potentials to add up because of the refractory period. But graded potentials can add up. So even just this one input, if you get 2 stimuli right in a row, can add up and take that second neuron to threshold. If it's only one neuron, adding up in a short period of time, we call that temporal summation, whereas in the previous example, that was spatial summation. So the overall picture then is that you have an action potential in one neuron. That triggers the release of neurotransmitter. Remember, an action potential never goes across the synapse for chemical synapses. The neurotransmitter crosses the synapse, triggers an electrical change in the postsynaptic cell, and if those electrical changes, either from that one neuron or many, add up, the second or postsynaptic neuron can have its own action potential, starting that cycle all over again. All right, I hope that was helpful. Let me know if you have questions.